Hi and welcome to this second part of the Integumentary System 1 lecture, and in this section we're going to be looking at the epidermis and the keratinocytes. So the learning objective for this section is that you should be able to list and describe the layers of the epidermis, including the major features of keratinocytes within each layer, and describe the process of keratinization. So let's begin. So of course, if you remember from the last video that the epidermis is a layer of stratified squamous keratinized epithelium, and of course that sits on a basement membrane. Now the epithelium, which we call the epidermis, can be subdivided into 4 to 5 layers depending upon whether you're looking at a section of thick skin or thin skin. Thin skin has 4 layers, thick skin has 5, and they're described from the base to the free surface as the stratum basale, which is the basal layer, the stratum spinosum, the stratum granulosum, and then the last of the layers, which is the dead cell layer, is the stratum corneum. Now in thick skin, there's a layer in between the stratum granulosum and the stratum corneum, and this is known as the stratum lucidum. As I mentioned, that's only visible in thick skin. So let's have a look at that. So here we've got a section of thick skin stained with hematoxin and eosin, H&E. We can tell it's thick skin for several reasons. Firstly, we've got this very large, thick stratum corneum layer. This is the dead cell layer, and you can actually see where it's been sloughing off or being abraded on the surface there. So that's very characteristic of thick skin. The other very characteristic feature of thick skin, and we'll talk about thick versus thin skin at the end of this lecture, we see this waviness associated with this epidermis, and these are actually dermatoglyphs or fingerprints, which are specifically localized to thick skin. So we talked about the epidermis starting off with the stratum basale, so labelled B here. The stratum basale is the single layer of cells sitting on the basement membrane. Then above those we have the stratum spinosum. This is the largest of the living cell layers. You can see it's quite a thick layer here. And then you can see why this is called the stratum granulosum, because the cells here have very basophilic granules associated within their cytoplasms, and so that's a very granular layer. These are the 3 living cell layers of the epidermis: stratum basale, stratum spinosum, stratum granulosum. The cells undergo cell death here and then form the first of the dead cell layers, which in the thick skin is the stratum lucidum. It's a translucent layer here sitting above the granulosum. And then above that we have the true dead cell layer, which is the stratum corneum. So there's no nuclei, no organelles. All the cells here are actually just membranes filled with keratin, so protein, and therefore that's why it's so cytophilic here. So the cells that make up the epidermis, well, the predominant cell type that makes up the epidermis, are known as keratinocytes. And they, of course, they begin in the stratum basale. The cells here undergo mitosis. One gets pushed into the stratum spinosum. One remains behind as a basal cell or a stem cell. And then the cells eventually migrate up through the layers of the spinosum, through the granulosum. They undergo cell death and then form the stratum lucidum, stratum corneum cell layers. So we can see quite a change in structure as the cells move from the basal layer to the spinosum, to the granulosum, and then eventually to the dead cell layers. So the basal cells, you can see they look quite cuboidal, or in some cases columnar even, in certain areas. So they start out this way, then they become this sort of more rounded, spiky looking cells. A bit difficult to see at this magnification, but I'll show you evidence of that in a little while. And then they become elongated granular cells, and then they undergo cell death. And then because they undergo cell death, we can't see their nuclei. It makes it very difficult for us to actually identify their shape properly, because we know we use the nucleus to identify their shape. But through electron microscopy, we know that these cells become very flattened, and some of these remaining stratum granulosum cells do appear very flattened as well. And we know that we characterize or classify epithelium based on the shape of the surface cells. And so despite the fact we can't see nuclei on the surface, we know what the final shape is, and so it's a stratified squamous keratinized epithelium. So the keratinocytes during their lifespan undergo a process called keratinization. So they start out as nice healthy living cells, they undergo mitosis, they move into the stratum spinosum and they make their way through those layers. And as they make their way through their layers, they begin to fill with these intermediate filaments called tonofilaments, and they begin to accumulate these granules called keratohyalin granules. Now, together the tonofilaments and the keratohyalin granules will form the final keratin, which we recognise as keratinocytes on the dead cells on the surface. And there's a third component that keratinocytes actually produce, and these are structures called lamellar bodies. Lamella means sort of a layer, and so it's sort of spherical layers of electron density associated with them, and these are actually a glycolipid. And so when the cells undergo cell death, the tonofilaments and the keratohyalin granules fuse to form the keratin, but the lamellar bodies are actually released from those cells and forms this protective glycolipid coating on the outside of each cell, and this provides a waterproofing layer on the outside surface. We know that the cells of the lucidum and corneum are dead cells, and they form these plates of keratin, and we know that's a good way to resist abrasion and also protect in conjunction with the lamellar bodies with waterproofing in a dry environment. So we're going to have a look at each of the layers of the epidermis in a little bit more detail, and we're going to start off with the stratum basale. So the stratum basale, of course, is the basal layer of the epidermis. It sits on the basement membrane, so it's the deepest of the layers in the epidermis, and these cells are described as being cuboidal or low columnar in shape. Now, that's their shape. That's not their classification, because we don't classify the sublayers. So they're all part of a larger epidermis, epithelium, which is a stratified squamous epithelium. These cells have a number of cell junctions, so they have a number of hemidesmosomes between themselves and the underlying basement membrane, and then that anchors the basal layer there, and then they also have a number of desmosomes between the cells, and I'll show you some electron micrographs of these in a little while. The cells may contain melanin granules. So melanin is a protective granule which comes to overlie the nucleus. So in this micrograph down on the bottom right hand side, you can see these sort of browny orange material within the cells. These are the melanin granules, and they protect the nuclei of these dividing cells from ultraviolet radiation. They're produced by a cell called a melanocyte, and we'll see that in this basal layer not only do we find keratinocytes, but we also find non-keratinocytes, and one of those non-keratinocytes that we find are melanocytes, indicated with the M here in this micrograph. The cells undergo mitosis in this region, so this allows for the constant production of keratinocytes as they migrate their way through the layers, and eventually they're sloughed off at the surface through abrasion. And as I just mentioned, we do find not only keratinocytes but also non-keratinocytes in this layer. So within the stratum basale, we can also find melanocytes, which we just talked about. They produce the melanin, which they pass on to the surrounding keratinocytes, and Merkel cells. And they're a special type of sensory receptor cell that allows us to detect touch, but we'll talk more about them in the next lecture, or a bit later in this lecture. So here's an electron micrograph I took. This is a section of skin from a human. So this is the basal layer. So these are cell keratinocytes within the stratum basale. You can see the nucleus of one here. Within its cytoplasm, you can see these what looks a little bit like hair within their cytoplasm. These are the tonofilaments, or the intermediate filaments. You can see the base of the cells here. This little line here represents the basal lamina, or part of the basal lamina there, and then this material out here, this is part of the reticular lamina. So together, the basal lamina and the reticular lamina form this larger structure, which is the basement membrane. So these are just a few fibroblasts in the loose connective tissue there. Now this is the intercellular space, and you can see some projections of the cells here, and then in some regions you can actually see some desmosomes or macular adherence between these cells. So you can see there's a nice one here showing these desmosomes between these cells. Oh, there's a really nice one there. Sorry, I just saw that and thought I'd point it out. So moving on then, the cells undergo mitosis. One stays behind as the stem cell, one moves out into the stratum spinosum, which is the spiny layer. So the spiny layer, or the prickle cell layer, you can see why they're named that. So in this picture here, you can see that the cells look like they've got lots of spikes on them. So lots of prickles, which is why they're called the prickle cell layer. And we'll talk about why that is in a little while. So they sit above the basal layer. This is the largest of the living cell layers of the epidermis. These cells are in constant contact with each other via a system of intercellular bridges. Basically, that just means they have extensions which join onto each other, like they're holding hands or something. Well, lots and lots of hands, obviously. And they're held together by desmosomes. Now, those desmosomes are really, really strong junctions between these intercellular bridges. And so when we process for microscopy, we know that one of the steps is a dehydration step. So the cells shrink a little bit, but those desmosomes are so strong that the intercellular bridges hold. And so we get some shrinkage between the cells, and what remains behind are these little spikes where the intercellular bridges are, and they're held together by the desmosomes. The cells begin to accumulate more tonofilaments, or intermediate filaments, and there's another type of non-keratinocyte which we can see. These are called Langerhans cells, and they're best identified within this layer. And there's none in this micrograph, but I'll point them out a bit later in the lecture, in another part of the lecture. So we're going to have a look at some electron micrographs now that I took. So this is a low magnified view of the stratum spinosum under electron microscopy. Now each of these little electron dense dots, or these little black dots, represents a desmosome. Okay, so you can see why we've got all these intercellular bridges held together by these desmosomes. So here's a magnified view. These are all keratinocytes, and their intercellular bridges are held together by these desmosomes. You can see how many of them there are just in this micrograph. And at highest magnification, we've got these desmosomes holding these intercellular bridges together. Now there's a really nice one here which is actually showing the tonofilaments anchoring into the desmosome here, remembering that desmosomes have intermediate filaments anchoring into them, which is why, partly the reason why they appear more electron dense than a zonular adherens. So moving up then into the stratum granulosum, this is the granular layer. These are responsible for the final living cell stage, the cells undergo cell death, so they produce eventually produce the surface keratin and the waterproofing substances. So the cells contain a number of basophilic keratohyalin granules, hence their name, the granular layer, abundant tonofilaments, and lamellar bodies. At cell death, the keratohyalin granules and the tonofilaments combine, and this helps to form the keratin. And at cell death, the other thing that happens is that these lamellar bodies are released, and they contain the glycolipids or glycophospholipids, and they help to waterproof the outer surfaces on this layer, and that just provides a protection from drying out. And because it's a lipid, it can be disrupted by detergents. So if you're someone, or you know someone who uses a lot of detergents, one of the consequences is that you remove this waterproofing protective layer, and you end up getting sort of cracked, dry skin, and that leads to potential for infection and things like that. So the last of the layers of the epidermis are, of course, the dead cell layers, the stratum lucidum and the stratum corneum. So these are, of course, dead cells filled just with keratin. There's no nuclei, there's no organelles, they're just protein, which is why it's quite acidophilic, or staining with eosin here. And we know that the surface keratin is constantly worn during normal wear and tear from surface friction. And of course, you can see that if you ever sort of get a nice view of it in the sun and you rub your arm, you can see those little specks floating around in the air afterwards, and that's the dead keratin cells. The other thing which you're probably aware of, but you may not be aware of, and you're probably going to hate me for telling you this, but most of the dust in your house is actually dead skin cells of you and your family and your dogs and your cats or whatever animals live with you. Those are dead skin cells on the surfaces of the place that you live. So you're welcome for me telling you that. So here's an electron micrograph. So here we've got the nucleus of one of these keratinocytes. You can see these very electron dense keratohyaline granules, this sort of more slightly paler but still relatively electron dense fibrous material. These are the tonofilaments. You can kind of see them sort of joining together there. The cells are undergoing cell death, so this nucleus is looking a bit more electron dense, which is typical of a dying nucleus. And then we've got the fusion of the keratohyaline granules and the tonofilaments. And then here's the stratum corneum. Have a look at this. It doesn't have any more organelles. It just sort of zips from here to here. I think it's just amazing. So there's the outline of the cells. You can see they're quite squamous, so they're flattened. No nuclei, no organelles, just this relatively amorphous electron density associated with all this keratin now filling these keratinocytes, these dead keratinocytes in the stratum corneum. So a keratinocyte takes about 25 to 30 days to move from the basal layer to eventually to be shed from the surface in areas with lots of friction, so the soles of the feet or the palms. In other areas of the body, it takes about 40 to 50 days for that process to the keratinocytes to make their way through the keratinization process and eventually be shed. Now, in some of these in disease states, such as psoriasis, this turnover period is much shorter, so the cells tend not to get as much time in the keratinized state, and so the person ends up with these very, thinned layers of epidermis and cracked, sore skin. And so this diagram here just shows sort of the process of keratinization. So we've got our basal cell with its hemidesmosome and desmosomes. It's got some tonofilaments here. They begin to accumulate these lamellar bodies, and eventually they'll form these keratohyaline granules which are fusing with the tonofilaments. At cell death, the cells discharge their lamellar bodies, forming this waterproofing protective layer, and then we've got our keratinized cells out here with no nuclei, just filled with this protein keratin. So that's it for this second part. Please now proceed to checkpoint number 2, and the quiz on, the quiz on Milo, and do the quiz, and then you can move on to part 3.